Yes, welcome back to my new class, in which we are talking about the genetical terminology. That is acquired character. The alternation in the morphology and physiology of the organism is responsible of its ecological factors. Environment is known as the acquired character. Acquired character are usually heritable. That means the organism that acquire the character from the environment it is known as acquired characters, or it is heritable also. Heritable, that means it moves to the generation to generation. Albinism, the absence of the color in skin, hair, eyes, and the absence of the chloroplast in plants and herited traits. That means the absence of color in the skin, यानी स्किन में कोई भी कलर नहीं प्रेजेंट होगा. Hair, eyes, or the absence of the chloroplast in the plant. Where like जैसे कि प्लांट्स में क्लोरोप्लास्ट नहीं होता तो प्लांट ग्रीन कलर के नहीं दिखाई देते. तो और उसी तरीके से अगर बॉडी में इनहेरिटेड ट्रेट नहीं होंगे तो उसे एल्बिनिज्म बोलेंगे. Albino. The animals without pigmentation in the skin, hair, eyes is called albino. When किसी भी एनिमल के अंदर किसी भी एनिमल के अंदर या फिर किसी भी ह्यूमन बीइंग के अंदर अगर कोई पिगमेंटेशन नहीं है, स्किन में कोई हेयर ब्लैक नहीं है, वाइट नहीं है, ब्राउन नहीं है, आइज़ का कलर येलो ब्राउन ऑलिव ग्रीन नहीं है, तो उसे एल्बिनो बोलेंगे. एलिल्स. एलिलो Allelomorph. One or two or more forms that can exist in the single gene locus, distinguished by their differing the effects of the phenotypic alleles, are gene controlling the same characters. For example, eye color, but producing the different effects. Example, black and red, and the occupying corresponding position of the homologous chromosomes. Alleles are those like the traits that are present on the chromosomes, and their phenotypically they are but their physical appearance is similar, but the genes are different because genes are transferred from one generation to next generation, but the phenotypic but the physical condition is same because no, sorry, physical condition is different, but the genes are remain heterologous, like heterologous chromosomes are present. Next, amniocentesis, the rupture of the uterine wall with the needle for the purposes of obtaining the amniotic fluid. Amniocentesis is the process in which the puncture of the uterine wall with the help of needle for the purpose of obtaining the amniotic fluid, which can be analyzed to determine whether the fetus has the genetic abnormalities. And the amniotic fluid contain the the sloth the sloth fetus cell. That means the amniocentesis is the process by which the person get identified whether the fetus is a boy or a girl. So this process is to be banned in many hospitals, and by this the organism by this we can detect that the organism is the fetus is abnormal or normal. Next, the Mendel and his work. Mendel and his work. Who is Mendel? Gregor John Mendel was a pioneer of classical genetics. He was born in July 22 in 1822 in Hesendorf in Austrian Silesia, where his father and Anton Mendel was the owner of a small farm. He went to one day he went to the farm where he saw there is little pea plant are sown in their farm, and these small pea plant having the flower is hermaphrodite because they have the pistil and stamen both. By the stamen, if there is no pollination occur at that time, he took the brush and put it on the stamen, and and found some anther some bushy and anther from that and put it into the pistil. By doing this, this is the pollination. But if the pollination occur at the same plant, that is called self-pollination. And if pollination occur at the different plant, then it is called the cross-pollination. But there is a man-made pollination, so this is called the self-pollination that occurs in the plant. By doing this, the pollination occurs and there is numerous anthers and anthers are formed during spores are formed during the process. By doing this, we get the new hybrid pea plant, that is known as, the scientific name of pea plant is Pisum stevium. Mendel selection of the experimental plant. The experimental plant having the Pisum stevium having the variations, reproduction, controlling mating, etc. Variation, that is, the organism has a variant. Some variation are there. That means if we take the normal pea plant, if we took the organisms which are able to choose in the genetical experiment should have the number of detectable differences at the time, only single detectable character should be considered. If we took the plant in which took the pea plant, the genetic experiment shows that the number of detectable differences at the time, only the single detectable character should be considered. जब वहाँ वहाँ पर बहुत सारे हमने नंबर ऑफ डिटेक्टेबल डिफरेंसेस निकाले, तो उसमें से कोई एक डिटेक्टेबल कैरेक्टर जो कि डोमिनेंट होगा उसको हम वहाँ पर कंसीडर करते हैं. दैट इज़ कॉल्ड द वेरिएशन. एंड इन द एवोल्यूशन this is a very important part that I'm talking to you, that the in evolution there is the dominant character that moves one generation to next generation, and this is known as the variation. And the organism when the dominant character moves to one generation to next generation, then this is known as the evolution. And if there is a in trait there is a drastic changes occur, then the process is known as mutation. And if there is lots of detectable characters should be taken, but only one single detectable character should be considered, then it is called variation. Next, reproduction. The chosen of the organism should be sexually reproduced by fusion of male and female gamete, the sexual cells, because only then the offspring can be able to receive the different characters from both male and female parents. We all know about the reproduction. Yes, reproduction means the fusion of male and female gametes. But the organism chosen show the sexual reproduction. यानी कि कुछ ऑर्गेनिज्म को हम लोग चूज करते हैं ताकि वहाँ पर क्या हो सके, फ्यूजन हो सके मेल और फीमेल गैमेट्स का. बिकॉज ओनली when the offspring can able to secure the different character from both male and female parents. बिकॉज जब हम किसी भी ऑफस्प्रिंग को देखते हैं तो क्या होता है, एक मेल और फीमेल कैरेक्टर ऑब्टेन करने के लिए एक एक्स क्रोमोसोम फीमेल पेरेंट से लिया जाता है और दूसरा एक्स क्रोमोसोम मेल पेरेंट से लिया जाता है. दैट मीन्स when they both X chromosomes are incorporate with each other, then we get the female baby. And if the X chromosome get from the mother side and Y chromosome is found from the parent side, then we get the boy boy as the offspring. And after that, controlling mating is the chosen organism should be able to mate in controlling the well planned condition. Because it is genetically experiment, sometime we have to be near about the genetically mating. That means controlling mating control करने का मतलब होता है that there is a there is a well planned condition in which genetical experiment should be taken. Genetically experiment should be occur, but we have to be control the mating, that we have there is a barrier between the male and female gametes, because so that they do not mate together. Next. Yes, Pure parents of methods of controlling mating. One should maintain the careful records of the offsprings by many generations. And short life cycle. The chosen organism should have very short life cycle. The chosen organism should have the very short life cycle. Those organisms should be taken for the genetical experiment. They have very short lifespan because they have very short lifespan because of their because of their detectable character, because of their dominant short life cycle. The chosen organism should have the short life cycle also, because it should be plucked from the plant and it has the dominant character, but it should be detectable part of the plant also. Large number of offsprings. Yes, the organism which have been chosen from the genetically experiment should be produce large number of offsprings after after each successive mating, because of it helped in the deducing the correct conclusion. Yes, large number of offsprings are produced during the genetical experiment. That means there is a fusion of male and female gametes without any barrier that should produce a large number of offspring each other the successive mating. In the every सक्सेसिव मीटिंग, बिकॉज इट हेल्प्स टू डिड्यूस द करेक्ट कंक्लूजन, बिकॉज वो वहाँ पर हमें एक करेक्ट कंक्लूजन तक पहुँचाता है. आगे है क्रोमोसोम्स. द साइटोलॉजिकल इन्वेस्टिगेशन have now established that the genes are the units of the DNA along with the RNA and the nucleoproteins and constitute the thread-like structure. Is called the chromosomes. Chromosomes are the cytologically investigated, the now established by the genes. That means कि cytological, the term cytological is related to chromosome, but but the DNA are along with the RNA. That means they are incorporate no DNA is along with the RNA with the nucleoprotein and constitute the thread-like structure, are stainable, is known as chromosome. And the chromosome having the centromeres and the two loops. The two loops having the traits or the genes on it. The genes like the parent chromosome parent chromosome, in which female chromosome and male chromosome. The female chromosome having the traits like height and the color of eyes, the color of skin, like this, and the same trait that is also found at the parent chromosome, but the parent chromosome at the male chromosome having and both the chromosomes it should be considered as who is dominant, and the one is dominant and other is recessive. The dominant character are shown in the offsprings, and the recessive characters must be present in the offspring, but do not act easily. For example, the blood groups as given by the Landsteiner, and Landsteiner explained that there is A blood group, AB blood group, B blood group, and O blood group. If AI, that means A is the dominant blood group, but I is the carrier. If A is crossed with I, then it forms A blood group. If A is crossed with B, that means AB blood group. If B is crossed with I, carrier, then it is B blood group. And if I and I are crossed together, they shows the O blood group. That means O is the recessive, and if O is recessive, then it is not shown easily, but O is the universal donor also, and O positive is the universal donor also, because it is recessive carrier. It is recessive and carrier also. Homologous chromosomes. What are the homologous chromosomes? The chromosome of the similar size and nature often form the pair during the meiotic cell division, and such identical chromosomes are called homologous chromosomes. Homologous chromosomes are very easy to understand that कि homologous chromosomes are those which are identical and they have show the cell division, that is meiotic cell division, which is also same present in both the chromosomes. So this is called homologous chromosome. Heterologous chromosomes are those which are different from each other. For example, the Drosophila, the fruit fly. The Drosophila having the male part, male is the vestigial wings, but the female has the large wings. If they are crossed together, they form the one is when they form the we are talking about the genotypic ratio. So there is a the long winged Drosophila, the long winged Drosophila, that is the long winged Drosophila, that is female, and the vestigial one is male, and then long one is the female, and the vestigial one is the male. That means this is called the heterologous chromosome. Incomplete dominance, the incomplete dominance in which the heterozygote dominance alleles are not the complete mask of the phenotypic expression of the recessive alleles, and they occur in the intermediate phenotypic of the heterozygote is known as incomplete dominance. Incomplete dominance is very easy to understand that with example, that is white hibiscus. Take a pluck white hibiscus and cross with the red hibiscus. If we cross with the red hibiscus, we found that there is no neither white hibiscus is found nor red hibiscus is found. Only pink hibiscus is formed. That means they are the incomplete dominance. And if we are talking about the phenotypic ratio, it is 3:1, but in the genotypic ratio there is 1:2:1. After that, we are going to start the codominance.